So the next speaker is also no stranger to Boston scientist. That's Bill Kaelin. So all three different Boston institutions are represented here. So Bill is from Dana-Farber Cancer Institute. So whereas David and I have been working on oncogene pathways, and Bill has focused on tumor suppressor pathways. And after doing really groundbreaking work with David Livingston on RB, he then shifted to studies of the VHL tumor suppressor and really enlightened the world on how VHL, the function of VHL and why it's a tumor suppressor, and that is that it regulates the destruction of the hypoxia sensing protein HIF by regulating prolyl hydroxylase, or regulating its modification by prolyl hydroxylases. So basically, we understand how oxygen is sensed through the studies, the really groundbreaking studies of Bill. And he's also worked on P73, member of the P53 family. So welcome to Bill to the podium.
Okay. Thank you very much, Joan, for that very nice introduction. It's a pleasure to be here. I have two disclosures. I do have a small financial interest in a company called FiberGem, which is developing small molecule inhibitors of the HIF prolyl hydroxylases, and I also consult for Agios, which is located nearby. You may know this is a company that is trying to develop new cancer drugs based on insights into cancer metabolism. My lab, as Joan mentioned, has had a longstanding interest in von Hippel-Lindau disease and the von Hippel-Lindau tumor suppressor gene, and here are the people who have done the work over the years. But really the work today I'm going to talk about was done primarily by Sungwoo Lee and by Julie Lossman. And we've also been aided immeasurably by many very generous and very talented collaborators. This is a partial list shown here, and I'll try to mention specific people as I go along, but I particularly want to highlight the contributions of Pepi Koivunen, who did a sabbatical in my lab, and my pointers, and also Ryan Looper, who's a terrific chemist out at the University of Utah. So as I mentioned, we've been working on the VHL tumor suppressor protein for many years, and you may know that the VHL protein is part of the ubiquitin ligase complex that in the presence of oxygen directly targets the alpha subunits of the HIF transcription factor for polyubiquination and destruction. You may know that HIF is sort of a master regulator of genes that promote adaptation to hypoxia, and it consists of an unstable alpha subunit and a stable beta subunit. The beta subunit is often called ARNT. And the instability, again, is shown, it reflects what I'm showing you here, that in the presence of oxygen, VHL targets HIF alpha for destruction, whereas when oxygen levels are low or the VHL protein is mutated, as often occurs, for example, in human kidney cancer, now HIF alpha can accumulate, dimerize with HIF beta, and activate those various and sundry HIF target genes. Now, the basis for the oxygen dependence was revealed by work we did about 10 years ago, and similar conclusions were reached by Peter Radcliffe, working at Oxford, namely that the interaction between VHL and HIF is oxygen dependent because HIF alpha has to be hydroxylated on one of two prolyl residues in order to be recognized by the VHL protein. Parenthetically, there's an antibody to this mark in the cell signaling catalog. So there's my gratuitous shout out to cell signaling. The enzymes that do the work here were originally called the Agonists family of prolyl hydroxylases, although they're sometimes referred to as the PhD prolyl hydroxylases. But by either name, what's important is that they have an obligatory requirement for oxygen. In fact, the oxygen atom of the hydroxyl group is derived from ambient oxygen. Furthermore, the oxygen KMs for these enzymes is just slightly above the oxygen concentration that would ever be achieved in a tissue. And as a result, as oxygen levels fall in a tissue, the rate of catalysis falls. And these enzymes also require, in addition to oxygen, reduced iron, as well as 2-oxyglutarate, also known as alpha-ketoglutarate, which then gets decarboxylated to succinate during the hydroxylation reaction. Now, generically, these enzymes belong to a larger superfamily of so-called 2-oxyglutarate-dependent dioxygenases that do similar chemistry and have interesting substrates we're going to talk about in a second. I will also point out that it's very clear that these enzymes can be inhibited with drug-like small organic molecules, many of which are acting as 2-oxyglutarate antagonists. So who are some of these enzymes? Well, it turns out there are a lot of these enzymes, and frankly, many of them you've never heard of because they've been either unstudied or understudied. And so, for example, here are the 3 enzymes that are capable of hydroxylating the HIF-alpha family members. But many of these other enzymes are, again, relatively poorly studied. An exception would be, many of you may remember that collagen has to be hydroxylated on proline in the endoplasmic reticulum in order to mature and form fibrils. And so really the collagen prolyl hydroxylases have been relatively well studied. More recently, pioneering work from Anjana Rao has taught us that the TET enzymes shown here are actually able of hydroxylating methylcytosine in DNA, and that's suspected of playing a role in the turnover of methylated DNA. And then the family got bigger still with the pioneering work of Yi Zhang at UNC and others who taught us that the so-called Jumanji-C histone demethylases are using a very similar chemistry. These enzymes hydroxylate the methyl group on the histone, which is then unstable and given off as formaldehyde. And so now you have this potentially intriguing connection where you could imagine metabolism through changes in things like 2-oxyglutarate or changes in oxygen could potentially affect chromatin structure through changes in histone methylation. And there's a small cottage industry trying to figure out which of these Jumanji-C proteins recognize which marks. And so, for example, you're going to hear later today from Jeff Settleman about a protein we had worked on years ago called RBP2 for retinoblastoma binding protein 2, also called JARD1A, now also called KDM5A. And with the help of Yi Zhang, we showed that this protein, in fact, is an enzyme which demethylates the H3K4 methylation mark. Now, in the cancer world, there's some interest in these proteins because many of these proteins turn out to be overexpressed in certain cancers, and yet other cancers are actually mutated. And so I would say at the moment, the champ would be UTX, which is mutationally inactivated in a wide variety of tumors. This recognizes the H3K27 methyl mark. So why did I give you that background? Well, it turns out, as you know, that Otto Warburg in the middle of the last century described this very peculiar metabolism that's characteristic of cancer cells, and he argued that the altered metabolism was a cause of cancer. But then over the next 70 or 80 years, the controversy was, well, is this altered metabolism causing cancer? Is it a consequence of cancer? Perhaps both, or perhaps neither. And so what Otto Warburg lacked was sort of a genetic smoking gun that, at least in certain circumstances, altered metabolism could clearly cause cancer. And so I would argue that's changed in the past 5 years because we now know that certain tumors have inactivating mutations of either succinate dehydrogenase or fumarate hydratase, which lead to the accumulation of succinate and fumarate respectively, which at least in a test tube can inhibit many of these enzymes. In fact, you might have noticed that succinate is the product of the reaction and can feedback inhibit these enzymes. And more recently still, we've learned that certain brain tumors and leukemias and now chondrosarcomas and now cholangiocarcinomas have peculiar mutations in isocitrate dehydrogenase 1 or 2, which allow these enzymes to produce millimolar quantities of a so-called oncometabolite, 2-hydroxyglutarate. And it's also clear that just as succinate and fumarate can inhibit these enzymes, 2-hydroxyglutarate is also capable of inhibiting these enzymes, at least in a test tube. So Bob Weinberg likes to say, just because it's a model doesn't mean it's wrong. A variation of that is, all models are wrong, but some are useful. So here's a sort of a model for how altered metabolism could, in principle, transform cells. Maybe, at least in the case of these mutations, you're transforming cells by modulating the behavior of one or more of these enzymes. So of course, the trick isn't to then show that my favorite enzyme in this family is inhibited in, for example, an SDH mutant tumor or an IDH mutant tumor. The trick is to say it's inhibited and it actually plays a causal role in transformation. So that's what I want to talk about today, and I want to focus particularly on the 2-HG. So it turns out there are two enantiomeric forms of 2-hydroxyglutarate. There's the R form and there's the S form, but in these IDH mutant tumors, it's the R form that's being produced. So we have to begin to understand how R2-HG can transform cells. So Pepe Karppinen in the lab, being a card-carrying enzymologist, began testing various purified enzymes to see if they could indeed be inhibited by R2-HG or S2-HG. And so here we have the collagen prolyl hydroxylase. We have the TET1 and TET2 enzymes that hydroxylate methylcytosine. And this is an enzyme that actually hydroxylates an asparagine residue. And in every case, she saw inhibition by 2-HG, but in every case, the S enantiomer is a more potent inhibitor than is the R enantiomer, and yet it's the R enantiomer that we're kind of worried about at the moment in terms of IDH mutant tumors. And similar results have been reported by others. Now, a particularly conspicuous example of this were the Kaelin prolyl hydroxylases that regulate HIF. Here, S is quite a good inhibitor of all three paralogs, Kaelin-1, Kaelin-2, and Kaelin-3, whereas the IC50 values for R2HG against these three enzymes is virtually immeasurable. And again, if you can read the fine print here, you may notice that we're looking at concentrations here that, for example, this is 1 millimolar, but it's been estimated that in tumors, the concentration of 2-HG might be anywhere from 5 to 30 millimolar. So even these sorts of IC50 values might be relevant in vivo. So at that point, Pepe asked, well, if R2HG is not an antagonist for these enzymes, could it possibly be an agonist? Now, this was somewhat heretical at the time. When we spoke to chemists about this idea, they either said, this is extremely improbable, to this is categorically impossible. So that was sort of a narrow space to be operating under or in. But nonetheless, undaunted, Pepe took recombinant Kaelin-1, Kaelin-2, or Kaelin-3, and she carried out hydroxylation assays in the presence of a recombinant HIF substrate. And on the y-axis, she's measuring the incorporation of hydroxyproline. So here we have no co-substrate added, namely no 2-OG, whereas in red or in olive, she's added the authentic co-substrate, namely 2-OG. But now in purple and aquamarine, she's adding, instead of 2-OG, R2HG. And R2HG can likewise stimulate this reaction. And this is specific because the S enantiomer doesn't do this. Now, again, this result was so unexpected, so heretical, I can tell you we did months and months and months of controls to try to make this result go away. We couldn't make it go away. So kicking and screaming, we had to decide that maybe it was right. I will say sort of the final experiment we did was with the help of Agios. They provided to us a uniformly labeled version of R2HG that we could then follow through the reaction. And it looks like the R2HG gets converted to 2-oxyglutarate, which is then decarboxylated to succinate. Now, fortunately, at the same time, Sungwoo Lee in the lab had been generating immortalized human astrocytes that he had infected with an empty retrovirus or a retrovirus encoding wild-type IDH1 or a retrovirus encoding IDH1 with this canonical R132H mutation that's been seen in multiple tumors and which we know allows this enzyme to now produce millimolar concentrations of R2HG. And in the interest of time, I'll tell you that indeed these cell lines do produce millimolar quantities of R2HG. And then as additional controls, he took these proteins and introduced additional mutations in their catalytic core so that these two proteins are now catalytically dead. He then grew the cells under normoxia or hypoxia and did Western blots for HIF1 and HIF2, as well as canonical HIF targets such as the GLUT1 glucose transporter. And what he found was that in the cells producing high levels of R2HG, HIF levels were in fact low, in keeping with the biochemical result I just showed you, namely that R2HG might actually stimulate the HIF prolyl hydroxylation reaction, which would then target HIF for destruction. So then you might say, okay, that's all right, but this is an overexpression study. How about real tumors or real cell lines with endogenous levels of mutant IDH? Now, it turns out, unfortunately, there aren't too many brain tumor lines that have endogenous IDH mutations, but there are a few. And so we obtained from Keith Lingen and Sam Weiss examples of oligodendroglioma cell lines that are either wild type for IDH1 or, again, have this canonical R132H mutation. And again, Western blots were performed in cells grown under normoxia or hypoxia, but you can again see that the HIF induction by hypoxia is severely blunted in the face of this IDH mutation. So that was encouraging, but you might say, well, but wait a minute, these are 3 cell lines from 3 different tumors, from 3 different patients, so they're clearly not isogenic. Maybe there are other genetic differences between these 3 cell lines in addition to the IDH status. And so at that point, we turned to Hai Yan, who's at Duke, and fortunately for us, Hai Yan had already taken HCT116 colorectal cancer cells and had used homologous recombination to knock into the endogenous IDH1 locus, this very same R132H mutation. And when Julie Lossman did Western blots on these cells, she again saw that HIF1 alpha induction by hypoxia was blunted when you had this IDH mutation. And finally, with the help of Roland Verhack, who was at the time at the Broad Institute, but who's now at MD Anderson, we looked at the expression of various HIF target genes in human brain tumors. And so along the y-axis here, you're looking at various canonical HIF targets. And of course, red means the transcript is up, and blue means the transcript is down. And since IDH mutations have been exclusively described in the so-called proneural subtype of glioma, and to try to compare apples to apples, we restricted our analysis to proneural tumors that were either mutant for IDH or wild type for IDH. And I think you can see that the HIF signature is indeed suppressed in the tumors that have an IDH mutation. So that all began to suggest that maybe there was something to this unexpected enzymatic result, namely that R2HG can stimulate the HIF prolyl hydroxylation reaction and can drive HIF levels down. But it, of course, begged the question of whether this had anything to do with transformation. So fortunately, Sung Woo had been characterizing those very same immortalized astrocytes I showed you a moment ago by way of Western blot analysis. And what Sung Woo noticed was that the cells that had the IDH mutation acquired the ability to grow in soft agar, which of course is a hallmark of transformation. So this then set the stage for him to ask, well, could this really be due to downregulation of HIF? And the answer appeared to be yes, because if he knocked down HIF-1 alpha with 3 different hairpins, in each case, like mutant IDH itself, this conferred the ability to grow in soft agar. And this result in turn was corroborated when, as an alternative way of driving down HIF levels, he simply overexpressed Agolin-1, and this was sufficient to promote soft agar growth. And this was specific because here's a catalytically dead Agolin-1 mutant that's incapable of promoting soft agar growth. So kicking and screaming, we came to the conclusion that downregulation of HIF, at least in this context, might be promoting transformation, or put another way, that at least in this context, HIF might be behaving as a tumor suppressor. Now, a lot of review articles point out why HIF might promote cancer in certain settings, and that's probably true, but there actually is an emerging literature that suggests that HIF could act as a tumor suppressor, at least in certain contexts. So this was work from Gabby Burgers in a mouse model where she noticed that if she deleted a HIF-1 alpha in astrocytes that were transformed, they grew more, they were able to go faster, and they were more invasive than their wild-type counterparts. And likewise, Peter Carmelite had published mouse data that suggested that HIF-2 alpha might likewise act as a tumor suppressor, at least in the context of brain tumor. So at least in the brain tumor context, we actually think that IDH transforms, at least in part, by lowering HIF levels, although we were open to the notion that it has many other targets as well. Now, Julie Lossman is a fellow in my laboratory, and she's actually trained as a hematologist with an interest in leukemia. So she took the very same expression constructs that Sung Woo had used to study astrocytes and now introduced them into TF1 erythroid leukemia cells. So again, acute myelogenous leukemia is another setting where one sees frequently these very same sorts of IDH mutations. So TF1 cells are useful because although they're leukemic, they remain dependent on cytokine for growth, and they can differentiate in response to certain cues, such as erythropoietin. And so here, again, Julie has infected TF1 cells with these same constructs that Sung Woo used a moment ago, but now she's going to ask the cells to grow in the absence of cytokine, and I think you can see that the cells expressing the mutant IDH can now grow in the absence of cytokine, and whereas the various controls cannot. Now, I mentioned another hallmark of these cells is they can be induced to differentiate in response to EPO. They become more erythroid, and that can be monitored by FACS staining for glycophorin A. So you see this nice rightward shift in cells expressing GFP as a control, or wild-type IDH1. You see this rightward shift indicative of differentiation. But in cells expressing this IDH1 mutant, now the cells don't shift. They don't undergo differentiation. Now, that's by a fax readout, but frankly, if someone has taken your space on the fax machine that day, all you really have to do is look at the bottom of the tubes, because these cells are redder than these cells, because these cells are undergoing erythroid differentiation. So at this point, Julie asked, well, is this really all due to R2HG? Because if it's really all due to this small organic molecule, R2HG, I should be able to just give the cells R2HG, and I should be able to transform them. So with the help of Ryan Looper, we obtained an esterified version of R2HG that gets into cells and gets trapped. And so here, Julie's treating the parental TF1 cells with increasing amounts of the esterified R2HG, and lo and behold, this actually is sufficient to confer cytokine independence. And this is enantiomerically specific because the S enantiomer doesn't do this at any concentration tested. Now, in contrast to the astrocytic system, however, at least in the TF1 system and in other hematopoietic cells we've looked at, knocking down HIF is not sufficient to transform the cell. So we were still operating under the assumption that R2HG was inhibiting one or more of these 2-oxyglutyrate-dependent dioxygenases, and that's how they acquired the ability to grow, for example, in the absence of cytokines, and that's why their differentiation was blocked. So at that point, Julie set up a very nice screen where she took her TF1 cells. She infected them with a pool of lentiviral shRNA vectors from the Broad Institute, targeting every known 2-oxyglutyrate-dependent dioxygenase. She then cultured these cells in the absence of GM-CSF, and she had identified vectors that were enriched over time by deep sequencing, the thought being that these would potentially identify for us enzymes that, when inhibited by R2HG, would in fact confer cytokine independence. So she got a number of positives out of the screen, and near the top of the list, but not at the top of the list, was the enzyme TET2, which I mentioned moments ago is an enzyme which we now know can hydroxylate methylcytosine. So it turns out 4 of the 5 hairpins in the collection were enriched over time, and we now know that this is simply an ineffective hairpin that doesn't knock down TET2. And this is very specific because, for example, if you look at the hairpins against the paralog TET1, there's no clear enrichment, so it looked like TET2 was a potential positive. This is just to show that these were true positives. So, for example, here are 2 independent hairpins against TET2. And as we had seen with mutant IDH itself, knocking down TET2 confers cytokine independence. And this is, again, specific because here are 2 hairpins against TET1. Now, at this point, I will tell you that although I described this somewhat elegant screen that Julie did, we had been thinking about TET2 for now almost 2 years because it had been shown by others that there were inactivating mutations in TET2 in acute leukemia. And so TET2 was the obvious choice to be the target. And to make matters worse, while we were doing this work, a paper appeared that said, yes, you guys, it really is TET2. We think it's TET2. And in this work by Ari Melnick and co-workers at Sloan Kettering, one of their arguments was based on the fact that they appreciated that in acute leukemia, mutations in TET2 and mutations in IDH are absolutely mutually exclusive, as though they accomplish the same thing. So that's a very nice and strong genetic argument for why TET2 would be the target. And they also provided some feasibility data that also suggested that TET2 was the target. The problem for us, however, and this was buried in a slide I showed you earlier, both S and R2HG inhibit TET2. In fact, if anything, S is a much better inhibitor by about an order of magnitude inhibitor of TET2. So this might have been an example of having done one too many experiments, because if we didn't have this experiment, we could have published something much earlier that would have been all very satisfying. But for a while, I sort of felt like the ancient mariner who had this result that we had to simply understand. And so Julie Lossman, who's very careful and thoughtful, went back to what I told you about earlier in my talk, which is the one set of enzymes where R and S appear to differ qualitatively, and not just quantitatively, are these Agolin prolyl hydroxylases, where, as I've told you, if anything, R is an agonist, whereas S is a fairly good antagonist. And so Julie wondered whether perhaps inhibition of Agolin was antithetical to transformation by mutant IDH or TET2 loss. And that appears to be the case based on the following types of experiments. So here is the result I showed you earlier. If you knock down TET2, you confer cytokine independence, as shown in green. If you now give the cells R2HG, if anything, you get even more proliferation, whereas if you give the cells S2HG, you inhibit proliferation. And I should also tell you, in the interest of time, that we've done the controls that show that both the R and the S get into cells equally, as best we can tell. And so at that point, Julie then obtained 4 different hairpins against Agolin-1 and took cells that had been transformed with mutant IDH, but now she could show if you knock down Agolin-1, with 4 different hairpins, you prevent the proliferation of these cells, in keeping with the notion that Agolin inhibition is antithetical to transformation in the system. And although there are 4 hairpins here, you might still, however, worry that maybe this could be some sinister sort of toxic effect, some off-target effect, because after all, this is a sort of a down screen or loss of function screen. So Julie married this type of work with assays where she asked whether knocking down Agolin-1 would actually restore the ability of her cells to undergo differentiation. And the answer was yes. So here we have cells expressing the mutant IDH. And now we come back in with a control hairpin, and as expected, when you give these cells EPO, you don't see this increase in glycophorin A staining. But now if she knocks down Agolin-1 with 2 different hairpins, now she fully restores the ability of these cells to differentiate in response to EPO. So the last question I want to address, and the one that's, of course, of greatest therapeutic relevance, is whether these effects are actually reversible, because now we have some tools that we can address this question. So one of the experiments that Julie did was to transform cells with the cell membrane permeable R2HG and then simply take it away. So again, here we're going to ask the cells to proliferate in the absence of cytokine. In red are cells which never saw the R2HG, and as expected, they don't really proliferate. In dark blue are cells that were continuously passaged in R2HG for at least 20 passages, and as I showed you earlier, they proliferate quite nicely. But now if you remove the R2HG for various amounts of time, you see that the cells do, over time, revert back to being normal, and they remember, if you will, how to become cytokine dependent. Now, fortunately for us, we've been also collaborating with Agios, which is developing IDH inhibitors, and they were kind enough to provide us with a tool compound that has been designed to specifically block the production of R2HG by mutant IDH1. And so Julie Lossman then performed an experiment where she looked at the proliferation of her mutant IDH1 cells treated with DMSO or treated with the IDH1 inhibitor, and as hoped for, the IDH1 inhibitor blocks the proliferation of these cells. So that's satisfying and sort of promising. But again, you could ask, is this simply some, again, sinister off-target effect, and we're getting fooled here? But fortunately, Julie can do the following controls. She can now say, if this is really through loss of R2HG production, then the cells I've transformed with R2HG itself should now be completely insensitive to the mutant IDH inhibitor, and that's the case. So here you see that the cells in dark blue, the vehicle-treated cells, are proliferating at about the same rate as the cells treated with the IDH mutant inhibitor. Likewise, in differentiation assays, if she takes leukemic cells that are expressing the mutant IDH, I told you earlier they're blunted in terms of their ability to differentiate in response to EPO, but now if she treats these cells with increasing amounts of the mutant IDH inhibitor, now the cells are able to fully differentiate in response to EPO, and she can do the same trick and show that this is specific because in the cells that she transformed with R2HG itself, now these cells are completely insensitive to the inhibitor. So we think in the course of this work, we've given some hope to the notion that mutant IDH might be a target in these IDH mutant leukemias and perhaps brain tumors. We've also, along the way, identified perhaps another therapeutic strategy, namely to think about the Agolins themselves as therapeutic targets. For those of you who have been trying to follow this literature, you may know that there are a number of papers that describe a number of epigenetic changes that occur in these tumors, and that may be true. But I think it remains an open question to what degree those epigenetic changes are actually causal for the transformed phenotype. At a minimum, there was already this growing fear that if IDH mutations operated by epigenetic reprogramming, that the effects wouldn't be reversible, or at least wouldn't be reversible in a clinically relevant timescale. I think our preclinical studies now give us hope that perhaps that latter conclusion is false and that some of these effects can be reversed. So we'll stop there, and thank you very much for your attention. I was very conscious, by the way, that you're all hungry and waiting for lunch. So if I was speaking even fast by my standards, it was because I was trying to get you to lunch.
Questions? Mike?
So, Bill, you said that 2-hydroxyglutarate, if I understood right, that it inhibited a variety of these, let me make sure I
Yes.
say this right, it inhibited a variety of the two OG-dependent dioxygenases,
Correct.
but it seems to specifically increase the activity of metabolic oxidation.
Yes.
Do you have any thoughts on the biochemical mechanism about how the same compound can take two sort of related enzymes and drive one down and one up?
Yes. The only thing I can say is some of our structural biology colleagues have modeled the interaction of R2HG and S2HG with the Agolin prolyl hydroxylases, and they can see, and even I can see this part, why categorically S would not be an agonist, and if anything would be an antagonist, whereas the structural data as it stands now leaves open the possibility that R could serve as an agonist because of the way it's interacting with the catalytic site, and in particular the iron. Okay? We don't know enough about how R2HG and S2HG inhibit the other enzymes mechanistically to address the other part of your question. So we think we're beginning to understand why you get these qualitatively different effects on the agolens. We don't know enough about the other targets. But again, the ability of these entities, R2HG and S2HG, to inhibit these other enzymes has been very well documented by a number of people now. So I think that result is real.
Those same structural studies point to any mechanisms by which mutation of the enzyme might be expected to lead to a gain of function?
Yeah, so the question is, the structural biologists, can they help us understand the gain of function? The short answer is yes. This is a little bit outside of my expertise, but you may know that, in fact, and Lou may want to chime in on this too. In fact, what the mutations, first of all, do is they let the reaction go in the opposite direction that it normally goes in. That's for sure. Secondly, there's some work that suggests that to really make R2HG, you actually want a heterodimer of one wild-type IDH and one mutant IDH, and the one then feeds the other, and you spit out the R2HG. And the last thing I will say is, very quickly, the word neomorph started appearing in the literature, that these mutations were neomorphic, but we're starting to learn from a number of groups, including Craig Thompson's, and we have similar data, that under certain specialized circumstances, even the wild-type enzyme can produce R2HG. Not to this millimolar level, but so what you're really doing is unmasking a latent ability of the enzyme to produce R2HG, and I'm not a smart enough structural biologist to really go any deeper than that in terms of your question.